Good morning. Welcome to the latest of AHDB's summer webinar offerings, Soil Health and Function in Arable Farming Systems. This webinar is doubling as an East Midlands regional webinar and also as our Northampton Monitor Farm summer meeting. No barbecue this year, unfortunately. I'm delighted to welcome our two speakers this morning. We have Rick Davies, Northampton Monitor Farmer, and Jamie Stotzka, Soil and Plant Health Specialist at Frontier, and I wish to thank them both for taking part. Before we do though, just a few bits of run through, and apologies to those who've already gone through this before several times. Just a reminder, you're all muted, which means we can't see or hear anyone. Any questions for the speakers, or if you're having technical issues, you'll see a box on the right-hand side of your screen. Can you type any messages in there, please? Questions will be saved for the question session at the end, but if you've got technical issues, Michelle Nuttall, who's coordinating all this in the background, we'll deal with that. We're due to finish about 10. We've also got a poll to add on to the end of that, so it might be slightly after. Basis and Noroso points should be available. We know that bases are. We're still waiting for Noroso to come back to us. Can you please make sure that you enter your name, postcode, date of birth, and your basis Noroso number in the question box? The webinar is going to be recorded and it will be added to the AHDB YouTube site. And this is our usual Twitter handle. If you have any feedback on the webinar or future webinar ideas, please let us know. It'll help us to shape the programme, and there's the opportunity for you to do that in the feedback at the end. At this point, I'm going to hand over to Rick, who's waiting there with his presentation ready. Over to you.
Morning all. Sorry about that, bit of technical issue. I hope everyone can see the screen okay. Please let Michelle know or type in a question if you can't. Anyway, this morning I'm just going to give a bit of a brief update as to where we are on the farm, bit of a virtual farm tour. Obviously we can't sit on a tractor and trailer and go around. So I'm just going to briefly go through where we are with the day-to-day on the farm, and then I'm going to pass over to Jamie, who's going to then run through soil health and function in arable farming systems. So, pretty high on the agenda for most people at the minute, but rainfall is quite a big part of our lives, and as you can currently see, there's our floodplain, which was in full flood in February this year. We've had in total 6 floods throughout the winter season, and you can see from there on, as predicted by a lot of people, that the rainfall then just fell off a cliff. So March was again a relatively dry month with a nice bit of rain just after drilling spring crops, and then as you can see, April and May, June have just been a bit of a non-starter. We've currently missed pretty much all of the rain in the last 10 days to 2 weeks. Big showers going past us, certainly last night, last Saturday and Sunday. So we are due a big rainfall event at some point, but we'll wait and see when it comes. Just going to go through farm sales currently. We have cleared out the sheds for 2019 harvest. So wheat, the last of the wheat went about 3 weeks ago. The oilseed rape, there was only 1 load of that anyway. And spring barley, spring beans both went last November. So as you can see, as my average prices for sales this year, wheat includes milling premium, which probably averaged in the main about £17 a tonne. So not my best year selling, but it's been quite a tricky one for all of us. 2020 sales, currently sold 4.5 tonnes a hectare of wheat which I think the last I sold was back in February, taking me to roughly sort of 45%. But given the current dry weather, that could easily be 60, 65%. And in hindsight, I wish I'd sold less, but that's where we are. Wheat fungicides, I pushed pretty hard through the hot period, getting up at 3 in the morning, going spraying, and then doing a bit in the evenings as well, in anticipation of heavy rain. It didn't rain, and then it came cooler and conditions were perfect. So you live and learn, but you've got to be on top of it. We're looking at possibly using some multi-N to top up proteins. We normally use ammonium nitrate as a last dressing, but with the lack of rainfall, I wasn't guaranteed that it was going to get into the crop. So we've got our local chap Andrew Caldwell from Frontier coming out tomorrow to do some more N testing, just to see where we are on chlorophyll and nitrogen levels in the crop, and then we'll make a decision then. Barley, I've just sprayed Earwatch this morning on the barley, so that's the door shut on the barley now. And the beans had a first fungicide yesterday at full flowering, and probably, depending on the season, we'll receive 1 or 2 more. As we sprang a couple of weeks ago, as the sun came up, it's the best time of day. I've been quite enjoying my early starts. It's nice to sort of get a couple hundred acres under your belt before 8 in the morning. It's been quite satisfying. Just thought I'd go back and just show you this. Some might be interested. We had a Claydon TerraBlade on demo in March, end of March, a little bit later than I would have liked. This was around about growth stage 31, which in an ideal world you'd want to be there in at 28 to 29. At the time it squashed a lot of crop, but it's recovered well, and you can hardly see where I've travelled. Really pleased with the results on some of the heavier land where there's a bit of moisture at the time. Some drier, brashier ground, it really struggled to penetrate and fill up any weeds. But we rode this field last week, and considering last year I couldn't rogue 8 hectares of it, and we ended up silaging some of it, we managed to rogue the whole 20-hectare field in about 3 hours. So really pleased with the results. Just going to whiz through some of our light land. We've got 2 main blocks. The home farm is 500 acres, and we've got a farm over near towards Bedford, which is pretty heavy. The home farm is predominantly sand and gravel with some seams of clay running down to silt on the river, on the floodplain, and then we've got some corn brash on the hills as well. So we've got a bit of everything. But you can just see here on the left-hand corner of the picture, wheat stands about mid shin, I suppose. Flag leaf's just curling up, and in places the top end of the ear has aborted. And you can see on the right-hand side there's a bit of a deeper drift. It's looking a lot better, but it's not looking well. And that's just to show you that's the picture of the aborted ears. I would say probably on the home farm we've probably got 20 to 25% looking like this, and then a further 25% looking pretty average, and then the remaining 50% is looking okay. But I'd say if we average 7 tons at home, we'll be doing bloody well, I think, this year. And let's just reiterate it from an aerial point of view, just showing the light areas there. Again, you can see where the cursor is, the mouse is. We've got it's called a cursor, which is 2,000 to 3,000 BC, which was in the news a couple of years ago. And there's a further one here to the left, which is a bit fainter, but it's the only good thing about this weather. It does bring out some historical features, but in an ideal world, you'd rather not see them. Again, this just confirms what I just said. If you can look at the imagery, we've got March on the right-hand side, which was the red end of the field, which you can see is down towards the river. This is the floodplain. There's a bit of flood damage caused back in February and March, where the crop was underwater or submerged for 4 to 5 days at a time. April, you can see the alluvial soil that came back well and started to tiller well. It recovered really well, and then now you can see in May on the left-hand side, there's a bit of a sand bank here, which is starting to go backwards, and green leaf area literally falling off. And then on top of the field, you can see the light patches as well. But, you know, it's just part of the season. Like I said, this is our heavy land first wheat. This is X days following spring beans, which has looked fantastic all the way through. I've been really impressed with X days. It's been clean as a whistle all the way through, and managed to make a few savings on fungicides. Very pleased with this at the minute, and looking pretty promising. So touch wood, we can get some rain, it'll finish it off nicely. As I touched on earlier, roguing blackgrass. We've been roguing now since 2012 when I came home to the family farm. So this is our 9th season of roguing, and so far our best. We managed to get across 250 hectares in just shy of 4 days with a team of 9 plus myself and another lad. So it was 11 in total, and we've managed to cover on a good day 80 to 90 hectares, and this year it's costing around about £16 a hectare. So really pleased with that, especially given the light that we only had on the majority of the farm. 1 hefty pre-em. Avadex was only applied on 50 hectares, no follow-ups, no peri or post-ems. So really pleased. Spring beans, it's not quite the best picture. I thought I had a higher resolution on this, but you can just see, again, spring beans this year on heavy land, following spring barley, where we flat lifted it in September, whizzed across with the Terrastar just to level it off, and then straight in with the drill. And it's been really effective, especially in a dry season. The roots have managed to get down, and the beans haven't really looked back. They're certainly knee to mid thigh. I'm really pleased with the height and the podding, the level of podding. And you just see here that in full flower, I've never seen so many flowers on a spring bean. So really pleased. And there's just a very early pod set at the minute. Explorer spring barley. This is down on the floodplain. So spring barley after spring barley going for seed. Drilled 375 seeds on the 15th of March. Went in well down here and has only received 40 mil, I think, roughly, in one hit, and that's all. So we've got about 17 hectares down the floodplain. Looks really well. And then my other spring barley is up on some heavy land, following stubble turnips, which were grazed back in December and Jan. The ground sat well originally, and then the very wet weather, February and March, really capped it, and it's sat pretty sad ever since. Unfortunately, these are the 4 dirtiest fields on the farm, and the idea of growing spring barley is to try and smother out blackgrass, but this is going to be a bit of a disaster this year. So I think we are planning on ploughing these 4 fields up here just to try and bury the problem and start again. So just a lesson learned: heavy land and grazing sheep doesn't always work. I've got my red poll cattle. I've only got about 20 at the minute, and this is in an old quarry down on some of the limestone. And as you can see, there's a little bit light on grass. They've got enough to go out for the minute. I've had a fair cut of silage early on, so it should be fine for a while. And then I just thought I'd finish on this, just showing the radar. It seems to be a common picture the last couple of weeks. No matter where the rain is, we seem to be either on it or in a corridor where there's no rain. For instance, last night the rain travelled east and west of us, heavy thunderstorms, giving up to half an inch to an inch, and we didn't even get a drop. So anyway, I shall leave it there. Any questions, you can either send them through to panel, or if you like, you can email me, and my address is there. Thank you very much for your time, I hope you enjoy the rest of the presentation.
Right, assuming you can all hear me, I'd like to say thank you very much, Rick, for that, for quick whiz through and very not long at all for what you managed to get over to us. At this point, we're ready to hand over to Jamie to talk about the soil health and function in arable farming systems. Thank you.
Thank you, Judith. Good morning, everyone. So I'm gonna try to whiz through quite a few things in as short a time as possible. And if there's any questions, obviously do ping them through, or also feel free to contact me afterwards. Judith, can we go to the next slide, please? So what I'm going to do is have a look at what actually makes a healthy soil. Obviously, soil health and soil function is a bit of a buzzword right now. You know, what the actual properties are behind that. In particular, I want to then focus on soil biomes, because I think the biological side of soil health is one that is still in need of the most exploration, and is actually, you know, much in focus at the moment. We'll look a little bit at soil organic matter, and throughout the presentation, I'll be highlighting assessments and tools that you could all use yourselves and or with your agronomists to have a look at how your soils are functioning. Next slide, please. So what is soil health and why does it matter to us? Next slide, please. So everybody's talking about it, like I said, and there are a few really, really good projects going on. For example, the Great Soils Project that is running through BBRO and AHDB, that from the frontier side, we also have the good luck to be involved in. You know, I think it's clear that we need to think about what we're doing with soils, because without soils, really, there isn't farming as we know it. But when we think about sustainability, for me, it's really, you know, those three areas: people, planet, profit. It's not just about the environment. It's also about making a living. And at the end of the day, everything that we look at has to fit into that for you. So we're looking at economical aspects, we're looking at the environmental aspect, and also at the legacy of what we leave behind. Next, please. And one of the going definitions at the moment of what soil health is, and which I think is probably the best definition right now, is that we're looking at a balance between the physical, chemical, and biological properties. So, you know, it's looking at different aspects of soil and how they all sit together, and how they can then form that basis for healthy crops and form that foundation for that. Next, please. And when we're looking at those three different areas, what it really means is within the chemical and physical arenas, that those will be areas that most of you will be quite familiar with, and will be familiar with testing already and assessing already. So within the chemical parameters, we're looking at things like pH, the nutrient content, and also organic matter. Within the physical aspects, we're looking at soil structure. Water balance, of course, has been very much in the forefront of everybody's mind all through this whole season, you know, really ending with last summer and then going into that wet autumn. And looking at the biological properties, that's anything from you know, earthworms, microbes, from the large to the small, but also the plants themselves really form a system of this biological system. Next, please. So what I want to do then is start looking at those three areas in detail, and looking at physical properties first of all. Like I said, this will probably be something that you'll be fairly familiar with. We're looking at aggregation, at structure. So how is the soil put together? How are the soil particles arranged? Clearly, having things like the biological system in there and having organic matter in there influences how aggregation and structure are set together. It's also things like surface sealing, compaction, and we've seen quite a lot of effects of the wet weather and then followed by the dry weather in those kinds of regards. So again, it's that kind of structural makeup of the soil. Next, please. It's also looking at the porosity. So when we think about how those particles are put together, and particularly how the different aggregates are set together, then that informs the air spaces, as it were. So we have small air spaces with micropores, and we have bigger ones, macropores, and that then shines a light on how water moves. So how much water can run through the soil, how much water is being held in the soil. So the more different size aggregates we have, and the more differentiated structure we have, the better that water movement and water holding capacity becomes, really. Next slide, please. And some of the things that you can do for yourselves are, for example, you can do VEST scores. There's some really good information available on that. There is a one-page A4 sheet from SRUC on how to do a VEST score. VEST stands for Visual Evaluation of Soil Structure, and basically just involves digging a hole and then looking at how the soil falls open and how the structure of the soil is made up. And I would encourage you to take a look at that, and take a look at that assessment sheet and have a go for yourselves, because it really tells you quite a lot about what's going on, particularly in problem areas. If you want to get a bit more fancy, you can look at things like penetrometer readings, which will show you areas of compaction. It will show you where those areas are. So if there are any pans in the soil, you will find those. Essentially, you'll also find out a little bit more about how dense that soil is, how tightly packed it is, or how open it is. And of course, if you're not into the DIY kind of testing, then there are commercial tests and reports that you could come back to, and you could get somebody else to do it for you and give you a full report on what's happening. And I think it's a good start to understand what's going on physically with that soil and how it is put together. And particularly, like I said, assess problem areas, but always compare them to the sort of best areas as well. So if you're looking back at yield maps, for example, look at where you have the parts of the field that are performing well and the parts of the field that aren't doing so well. And you can also compare that with undisturbed areas like under hedgerows or under pylons, where the soil is really left in a natural state and should give you a bit of a benchmark of how good it could be if nothing happened to it, as it were. Next, please. So going into the chemical properties then, that is, like I said, looking at things like pH, soluble salts, nutrient holding capacity, and the actual nutrients that are in there. Now, I'd wager a guess and say you're probably not going to do this at home. I do know some people who do, but I think the best way forward is to send off some samples to a lab and have those things fed back to you. But clearly understanding what the nutrient content in your soil is very, very important for how you grow crops. And I would say probably most people do those kinds of tests on a very regular basis already, and you'll already be adjusting your pH to make that nutrient to make the nutrients available, you know, at an optimum level as much as possible. Next, please. Particularly on organic matter, there's a couple of different methods of looking at that. So I think that is a very big part of the sort of chemical makeup of the soil, and I'll go into that in more detail in just a moment. Like I said, pH tests you'll already be doing. There are home kits, but it's probably a lot more reliable to have these sent off, particularly for nutrient analyses as well, because some of these parameters aren't so easy to measure, obviously. So I would encourage you for a lot of these to send them off to a lab, but choose a reputable provider and always stick to one lab. What we find is that with a lot of tests, you know, that the methods might be slightly different, but if you actually stick to the same lab, even if there is slight variation between different labs, it gives you a good benchmark of what's going on, particularly with the organic matter. I would say there's a couple of very, very good labs in the UK, and choose one of them and then stick with that. Next, please. So looking at organic matter, I want to talk about what it is and how it builds up, and how to measure it. So organic matter really refers to any material that contains carbon and that was formed by a living organism. So whether that's crop residues or dead organisms or manures, cover crops, anything like that, compost, anything that you either put into the soil or that degrades in the soil as an organic material from something that was there before, even including a crop, like I said. Next, please. And what it does is it serves as a reservoir for nutrients. It serves as a reservoir for water as well. It helps with reducing compaction, surface crusting, so it really aids the structure of the soil and helps with water infiltration and drainage. It's almost like having a sponge buried in your soil, I would say. So if you think about what that would do, how it gives you that more open structure, and it holds soil, but also, it holds water, but also the water can run through. I like to think of it like that because it gives me the best kind of idea of what it would look like as a picture, as it were. Next, please. And if we look at the sort of the life cycle, as it were, of soil organic matter and organic material, what happens is that we put in an organic material, like, for example, a manure, and that then needs to be broken down. And it's broken down, first of all, by the meso and macrofauna, so by the big life forms, as it were, things like earthworms, and that breaks it down to bigger particles. Then we end up with smaller particles, and the simple compounds in that are broken down by fungi and bacteria, by a specific set of fungi and bacteria that are called saprotrophic, and they break these down enzymatically, and it's fairly generalist at that point in time. But then we have further breakdown, further decomposition beyond that by very specialist microbes. They produce things like waxes and phenols, and they can help to break these materials down into more stable forms. And when we get to the most stable form, that's called humus, that takes many, many years, sometimes centuries, to really form. But anywhere within that journey, the nutrients become available to crops. And also that kind of physical structure being added into the soil gives us those benefits throughout. Next, please. So in terms of assessing organic matter, at the moment there are two methods that are most widely used, and I just wanted to talk about them quickly because there is sometimes some confusion about the readings that we get back when we have one of these soil tests done. So the two assessments that are done are loss on ignition, or LOI, and the combustion method, which is named after Mr. Dumas. So LOI measures the organic matter, and the combustion method measures the organic carbon. So there's a slight difference in the two of them. When we look at loss on ignition that's done at a relatively lower temperature in comparison to combustion, so the material is ashed. There isn't one protocol that's used all around the world. So each lab will have its own protocol for this. So that it can be anywhere between 350 and 650 degrees C. What we do is basically then burn off the organic material in the sample, and by measuring the weight before and after, we then know the fraction of organic material that's been lost. Now, one of the problems with this particular assessment is that when we have calcareous soil, sometimes the carbonates in that read to falsely high readings. So basically they're burnt off and then they're measured as organic material when really it was carbonates that we lost, as it were. Likewise, some clays can hold some structural water that, even though we dried out the soil before measuring it, still then sits in those clay particles and is evaporated and is also read as a falsely high reading. So that's why when you get back your assessment and you see that sometimes the loss on ignition will come in with much higher values than in the organic matter than the Dumas method. And that's why it may have picked up on something that wasn't actually organic material. The Dumas method is an oxidation process, so the sample is combusted at around 1000 degrees Celsius. It's a much more clearly defined protocol, and it gives us a reading of total carbon. So we look at the CO2 that's coming off through that oxidation process. So you'll have on your report, you'll usually have two readings. You'll have the carbon, and then you'll also have the organic matter. And in order to arrive at the organic matter, typically that's multiplied by a factor of 1.74. although there is some discussion around whether that's adequate or whether that should be a factor of 2. So watch out for that. It might change in future. But that gives you then how carbon is converted to organic matter. Okay, going on to biology then. Next, please.
Could I just interrupt you there, Jamie, before we move on to the next bit? A couple of questions have come through, and you've been talking around the right area, but just to be sure, first one, is there a measurable difference between different types of organic matter? It often gets termed organic matter, but I guess it's essentially soil carbon. Presumably there are differences between long-term and short-term.
Absolutely, there are many different types of organic matter, and that's one of the things that we're quite limited in at the moment. So what we're measuring here is a quantitative measure, not a qualitative measure. It's absolutely right. There are things like, for example, light fraction organic matter tests that aren't included in standard tests. So it is fairly limited what we're looking at here, and indeed we're really just looking at carbon in a way, and not at what type of organic material or organic matter, at what type of or at what point of the life cycle of the organic matter we are at this point in time. So I think, you know, in the future we will need probably more differentiated assessment methods for this.
Very good. And this second question, what's the difference between organic matter and humus?
So humus is the stable form of organic matter. So if you think about organic material going into the soil, then being cycled into what we would refer to organic matter, where it's a broken down form of that. And once it reaches that completely stable form where it cannot be broken down anymore, that's what we refer to as humus. And that can take, like I said, millennia to form. Okay, so moving on then to biological properties, what I'd like to do is break that down into a few different areas. So I'm going to start off with the big guys. So we're starting off with macrofauna, the type of animal life that we can see and that we can observe. Going to meso and microfauna, and then into microorganisms. And I will have a look at, in particular, a couple of very interesting microorganisms, or types of microorganisms in there that are symbiotic. But part of the system, like I said, is also the plant itself, so roots themselves, and you know, having that organic matter in there is the food that keeps it going. It's the fuel that keeps that whole system going. So all of that forms part of the biological property of the soil and its activity, how it all works as one system. Next, please. In terms of assessing biological properties, so if we already saw that assessing organic material is quite difficult, it becomes, you know, a lot more complex when we look at the biological properties. But there are a few things that we can do, starting off with worm counts. And when you're doing that, you're going right to the top of the food chain in a way. So if you know that you have a fairly decent amount of worms, and you should get something, a very good score would be about 16 worms per spit. So every time you dig up 1 spade's worth of soil, if you find 16 worms from different types in there, then you're doing very well. There are some good information sheets on that out there, including the AHDB sheet, which I think we will tag on to this at the end, so you can download that for yourself. There are also other tests like the CO2 burst test or the Solvita test, which I will talk about in more detail in a moment. And then there's more specialist analyses like DNA scans, which are a bit sort of in development at the moment, but my hope is that we will get something more usable fairly soon over the next few years, and that should give us a much more differentiated way of looking at soils going forward. Next, please. So the first thing I want to talk about before I go into more detail on the organisms themselves is this CO2 burst test. I would think probably quite a lot of people have come across it before. A lot of standard soil tests now include this. Solvita is the brand name, so that, you know, that's what it does. It looks at the CO2 that's given off by a sample of soil. So essentially the sample is dried and then re-wetted in the lab, and then we stick this little paddle in a jar and measure the amount of CO2 that's given off. So we're measuring the amount of respiration that's happening in the soil. And what that does is it tells us a total, as it were, again, it gives us a quantitative feedback of microorganisms in the soil. What it does not tell us is a qualitative measure, so we don't really know who's breathing. So if we imagine we could all get together in a room and we would seal that room off, and we would then measure the amount of CO2, it would give us an idea that there are some life forms in the room, but we wouldn't know necessarily what type of life form. So if we all brought our pets and we all had our dogs and cats in there, we wouldn't know who's a person and who's a dog. And this is similar to that. So, you know, whilst we do know that something is going on, we're not entirely sure what it is. But at the moment, it's kind of the best that we have until we have more differentiated and affordable differentiated methods like DNA tests. And I do think it's a good indicator. And again, it helps to benchmark how much microbial activity is happening in the soil at this point in time. Next, please. So looking at soil life then, what we'll do is look at things from the visible to the microscopic. And starting off with the visible, the macrofauna, the large living animal like kind of life forms that we have in there, we think of those as the structural engineers as well. So what they do is they move around a lot of soil, they move around a lot of material, they allow for root penetration because of that, they improve aeration, water infiltration, and they start breaking down that organic material and they start incorporating that into the soil so that that things further down the food chain, the smaller kind of organisms can start feeding off of that. So this can include things like mammals, worms, insects, millipedes, anything that we can sort of see that lives in and on and around the soil. Next, please. And in particular, one type of organism to focus on there are earthworms. We know that they do a great number of different things for us that are very beneficial. We have 3 main types of earthworms and up to 10 common species in the UK. The 3 main types are the epigeic, that are the litter dwelling, quite small worms that we find on the surface. The endogeic type of worm, which is the topsoil worms, they're the ones that we are probably most familiar with, you know, the ones that we see most often when it rains. They come out and crawl from one part of the field to another. And the anisic, the deep burrowing worms that are much larger than the others and that tend to live up to 2 meters in depth, actually. And they also build what's called middens. So for those of you who aren't inclined to dig for worms, then one thing you can do is count middens, where you'll find little piles of worm casts mixed with rocks and crop litter and so on. They pull to the top of the burrow, and that gives an indication of that particular type of worm and how many there are. What's important to think about is that both number and diversity are important, because as you can see, they live in different areas of the soil and therefore they do different things. So they provide different services. Next, please. So the way that that earthworms help to improve plant productivity is because, first of all, they help with nutrient mobilization. Like I said, they start breaking down those organic materials and making nutrients available. And in that sense, they also help with carbon cycling and supporting microbes. They also help with water infiltration simply by making a burrow. So they just make a hole in the soil and they move around soil structure. Likewise, they also help with soil aggregation, and this falls into something that we refer to as the sticky stringback theory, which I will come back later. But building aggregation in the soil and helping with that overall makeup of the soil and the water infiltration and water holding capacity. And in a greater environmental context, of course, none of these things live by themselves, as it were. They are all part of that greater ecological system that they live in, and they also function as a food source for native birds. I know some people that are quite heavily invested in stewardship schemes and things like that. It actually is quite interesting to think about that. Next, please. Going into the meso and microfauna then. These are sort of sitting in the middle between the larger organisms and the micro organisms, the microbes, and they are very interesting they tend to not be focused on that much, because essentially we're already starting to need a microscope to see them, and for the most part you need to be an expert to actually identify what they are. However, it is important to know that they are there. I think if we know that we've got good earthworm numbers and we know that we have good microbial activity, then we can fairly reliably think that we probably have a fairly good and diverse population going on in these particular areas. So within the mesofauna, we have things like mites and springtails. They break down those decaying organic materials further. And within the microfauna, we also have things like protozoa, nematodes, and so on. What's important to remember here is, people hear nematodes and they usually think, oh, I don't like those because I'm trying to grow potatoes or sugar beet or whatever. And I think what you can all see here is we're kind of getting into the territory of the good, the bad, and the ugly. So when it comes to earthworms, I think we can say ubiquitously that they're just useful and helpful. Whereas when we get into these more complex systems here, there is, of course, more beneficial types of organisms, and there is more unhelpful types of organisms for what we're trying to do. But they also all feed off of one another, and they all work together in some sense. So what we try to do is shift towards the more beneficial within these areas, but we probably have to take into account that we are always going to have a balance between all kinds of different organisms within this. Next, please. Going into microorganisms, then, that includes things like bacteria, fungi, viruses. So again, good, bad, and ugly all together. And there's a vast variety of functions, from, again, working with soil stability, aggregate formation, helping again with that organic matter, nutrients, nutrient uptake. But also there's a lot of very intimate functions that happen together with the plant, where, for example, the presence of some of these microbes triggers induced systemic resistance and helps with plant growth promotion. They produce antibiotics, hormones, all kinds of useful services coming from this type, this type of microbe and this type of specific microorganisms. Next, please. And within that, there's a specific set of microbes that are very interesting, and they are symbiotic. And symbiosis really just refers to living together. And one of the symbiotic relationships that we'll all be most familiar with are bees and flowers. So we're just looking at a close and long-term biological interaction between two different biological organisms. And in this case, we're looking at a microbe and the plant. Next, please. And the first of those organisms that I want to look at are the mycorrhiza. And what they do is, in that symbiosis, they exchange nutrients and water for organic carbon from the plant. So both of them get something good out of this deal, as it were. Next, please. Mycorrhiza refers to the actual symbiotic association. So it refers to the fungus and the plant together. So it's a mycorrhizal fungus plus a plant makes one mycorrhiza. Mycorrhiza is a bit of a hybrid term from Latin and Greek, meaning fungal root. And this is an obligate mutualistic symbiosis. That sounds a bit complicated, but all it means is mutualistic refers to the fact that both get a good deal out of it. So there's an equal exchange of benefits. An obligate here refers to the fact that the fungus has to have a host plant to survive. And that's quite important to remember in arable systems, actually. It's a very old symbiotic relationship. We know that it's been in existence for about 460 million years. So I always think that's the best sales pitch for anything in nature. And it's been around that long. But some plants have opted out over the course of evolution. For example, the Brassicaceae, like oilseed rape, and the Amaranthaceae, that's the beet family. So sugar beet is non-mycorrhizal as well. So again, that's important to remember when we think about the fact that that the fungus has to have a host plant to survive. The plant can do without the fungus, but the fungus cannot live without the plant. Next, please. The benefits are quite wide-ranging again, but one of the first and most obvious benefits is that the fungus itself grows into the plant root and then out into the soil, and it extends the surface uptake area up to 1,000 times. So we can get a lot more water and nutrient to the plant. The fungi also solubilize phosphorus by secreting acids into the soil, so they unlock chemically bound phosphorus. And then there is plant internal responses that are switched on, like better internal water use efficiency and also systemic resistance. And they help with soil aggregation, and again, I will come back into that in just a moment. Next, please. The next type of organism I've just got a couple of slides on is the PGPR, or the plant growth promoting rhizobacteria, also sometimes just PGPB, so that's plant growth promoting bacteria. A lot of these are endophytic, so they will live in the soil, but also in the plant tissue. So anything that lives in the plant tissue we refer to as an endophyte. And you'll be most familiar with that type of relationship from legumes in the rhizobium that live in legumes. There are 4 different mechanisms that help here with growth promotion. So we get biofertilization. Again, we get solubilization of phosphorus through these organisms, but also a degree of nitrogen fixation is possible with some of the PGPR. Get stimulation of root growth. That happens usually through hormone production, but also having more nutrient available to the plant. They can help with rhizoremediation, so the breakdown of toxins, and they help with plant stress control as well. Next, please. So looking at the list of benefits, that again explains how these different effects are achieved. So again, like I said, we get that nutrient to the plant. Plant growth hormones that are being produced by the bacteria help with that crop production. Systemic resistance comes back to induced systemic resistance, that is essentially an upregulation of the plant immune system, as it were. So having the bacteria there triggers specific genetic responses in the plant and helps it to defend itself against biotic and abiotic stresses. They also improve water use efficiency. Again, that's an internal reaction of the plant and can help to compete against pathogens. So that's coming back to what I said earlier on about shifting the balance of the microbiome into something more beneficial. And we know that they work very well with arbuscular mycorrhizal fungi. So when we think about that biodiversity, the more of these beneficials we have, the better it gets, because they all work together. Okay, then if we move on, unless there are any questions at this point, I'd like to look at how it all connects. So looking at what biology does then in terms of the other 2 aspects, in terms of the chemical and the structural. The first link I'd like to make is the link to structure. And one of the things that we know that mycorrhizal fungi do, in particular arbuscular mycorrhizal fungi, is they produce a sticky protein that's called glomalin. And that helps with the formation of aggregates. What you see there in those pictures is a fluorescent dye applied to glomalin, and you can see that it surrounds that root, the mycorrhizal fungi. They produce this to protect themselves, as it were, as a protective sheath. But it then also surrounds soil particles and binds them together into aggregates. Next, please. And that is part of the sticky stringbag theory. So if you think about the fungal network itself, the fungal hyphae, which are the body of the fungus, growing around those soil particles and through that soil structure, which is then also made sticky, then that holds those aggregates and forms bigger and larger aggregates in the soil. It's also helped by, for example, worms and other organisms that also make these sticky proteins. So it's not just the mycorrhizal fungi, it's also others. But having the combination of the fungal network together with the sticky proteins really, really helps with that aggregation. So I think that is one of the most important links between biology and the other aspects of soil there. Next, please. And linking biology to nutrients, we've already touched on most of this, but clearly we can get more nutrients into the soil, or not into the, well, yes, into the soil in terms of nitrogen fixation, as it were, but also we can make more nutrient available to crop plants via phosphorus solubilization, for example. And then there is that transport, that additional transport via mycorrhizal fungi, where we can reach more areas off the soil and more efficiently transport particularly phosphorus to the plant. Next, please. And then finally, looking at the link to organic matter. And I already talked about the sort of life cycle, as it were, of organic matter. But you know, that's being passed down that whole food chain, and then, as I said earlier on, it eventually becomes stable. It forms humus. But as that's happening, it feeds the biological network, and it serves as a nutrient reserve both for the biology but also the crop plant. So again, if we think about the crop actually being part of that biological system, it feeds that entire system. And that's how all of that then links together and forms one big healthy system, as it were. And onto the last slide, please. So I hope that has given you an overview of how everything hangs together, as it were. So I hope that it gives you an idea of the different aspects of soil function, of soil health, and a better understanding of what we really mean when we're talking about that. How to find out what's going on in your soil, you know, whether it is something that you can do yourself, or whether that's something that is best handed over to a lab or a specialist service. And then how, you know, having an idea and benchmarking all of that, and then working with improving those systems, working on improving organic material. I know that Rick, for example, uses a lot of organic manures and things like that to improve the system, how that can help feed the system, and how it can help to improve resilience both in the soil and then consequently in the crop itself. Happy to take any questions on that. And like I said, if we run out of time on this, also I'm more than happy to field questions afterwards if you want to get in touch with me. Thank you very much.
Thank you, Jamie. We do have some questions. I'll not particularly in any order, but this one's probably relevant to the last bit you were doing to some extent. What effect does flooding have on the micro and macro levels?
So flooding will, of course, alter the biome quite radically at times, and it depends on how persistent it is and how long the soil is flooded. So you'll already all have an understanding of the shift, for example, through the nitrogen assimilation cycle, where we're going from aerobic to anaerobic bacteria. And that happens in terms of the microorganisms in the soil in general. Some things are more resilient to flooding. For example, mycorrhizal fungi actually don't seem to mind. So they are found even in rice paddies. And once the flood is gone, they tend to recover fairly quickly. Likewise, earthworms seem to be fairly resilient. They can actually survive fully submerged for a few weeks. And they'll also find little nooks and crannies in the soil where they can survive. But it's the bacterial balance that tends to shift, you know, in waterlogged conditions. But because bacteria also have a very quick life cycle, we then see that shift back as soon as the soil is drained as well. So it doesn't really have a very lasting effect, I would say. So I would wager a guess that anybody who was completely waterlogged earlier this year, by now their soils would have shifted back to something quite different again.
Okay, thanks. And here's another one. What's the effect of chemical fertilizers and pesticides on soil health in general? And at what point do these chemicals begin to have an effect on the soil?
So it's a bit, I don't want to be too generalist, because obviously different effects are seen on different organisms. And as you can imagine, with something as complex as this, I can't really say all fertilizers affect this part of the system, or all pesticides are particularly harmful to another part. I personally did some studies around mycorrhizal fungi. And there are some pesticides, for example, that aren't particularly helpful for mycorrhizal fungi. But in general, we found that a lot of the chemicals used in arable farming seem fairly safe. Soil applied pesticides are much more of a problem. And some soil applied pesticides clearly wipe out just about everything. So if we're putting in a nematicide, then that's going to wipe out all the goodies as well as the baddies. So soil applied pesticides always will be a much bigger problem than foliar applied. In terms of fertilizers, I don't think that the levels of fertilizer that we apply in arable farming will have a hugely detrimental effect. There is some information about mycorrhizal fungi functioning less or colonizing less in high phosphorus environments, for example. But in my opinion, and from what I have seen in the research that we've done where I worked in the past, it would take a very, very high level of nutrient to actually really impact on that system. So that would be more looking at very intense systems like fruit production systems and closed systems like that.
Okay, thanks. This one's got some numbers in. You might need to jot them down. It's fairly specific. What can be done to improve soil health on fields with reasonable organic matter levels? 2 soil health samples, both around 4% organic matter, but one with soil respiration of 119, soil health score 65, the other a respiration of 148, soil health score it's gone, soil health score of 83. Did you get that?
Mm-hmm. Yeah, that's fine. Basically, yeah. What it tells you is that in the one soil with the higher respiration rate and the higher soil health score, essentially you've got more biological activity, you've got more microbial activity. What the microbes do is they process the organic material more quickly. So in a sense, it's good because you're cycling more nutrient back to the crop more quickly, but it also means that you're breaking down that organic material more quickly. So you need to put more into the system, as it were. So where you're trying to keep some of the sort of half digested organic material to open up that structure and have that spongy kind of nature. For example, in a sandy soil that's very open, that gets broken down more quickly, because the microbes can get to all of it more quickly, and that's why we lose organic material so much more quickly. So it's kind of a good news, bad news scenario. So in your in the sample that came back as having that higher respiration rate and higher activity, you are feeding that organic material back to the crop as nutrient more rapidly, whereas in the other part where you've got the lower respiration rate and the lower microbial activity, that breakdown is slower, essentially. So I mean, you would need to know more about the field to see, you know, what it looks like structurally and whether there's any organic material just sitting on top clogging up the system, for example, to really assess this and really take into consideration all of the different aspects around this. But that's basically what's happening here. So if you're looking at increasing your organic material, the one that comes back with the higher respiration rate for organic matter, you'd probably have to put in more material more often, essentially, as long as it stays active like that.
Okay, thanks, Jamie. Now, we have more questions. I don't think we're going to have time to do every single one, but we will follow them up afterwards if we don't get time. At this point, Rick has a question. Would you like to come in there, please, Rick?
Yeah, hi, Jamie. It was just regarding nematodes. Obviously, in root cropping, people often test for nematodes, obviously non-beneficial nematodes. Is there a good test for finding out beneficial nematodes in cereal cropping, and also extending
beneficial.
that to... Sorry,
did you say beneficial nematodes?
Yes, beneficial, yeah.
Okay, and sorry, and also extending that to...
Just is there a best test that you found for mycorrhizal activity?
Okay, so for both of them, actually, they're fairly specialist tests, unfortunately. So either you have to know how to visually assess, or you need to do a DNA test to find out about either of those organisms, as it were. So it's unfortunately not really a DIY kind of thing. At the moment, you'd have to find specialist labs to do either of them. For mycorrhizal fungi, I'm only aware of one service that does it at the moment, and they're fairly limited because you have to take root samples, stain them, assess them under the microscope. So at this point in time, I'm holding out hope for affordable and easy to read DNA tests. There is something there's a bit of a ray of light at the horizon. So we think that there are some services now coming around that will be able to give us that information, and also give us information about that in the context of the greater biological system, and not just looking at those specific organisms. But yeah, unfortunately at the moment, yeah, that's a bit of a, you have to send samples off to a lab, I'm afraid. It's not really a DIY thing.
Okay, thank you.
Now then, we'll just let Jamie have a rest for a few seconds, because I've got a question for you, Rick, that's come through. Jamie, I'll probably just quickly select 3 more questions after this one, but this one's for Rick, so you have a rest and a drink. Rick, what practices are you using to improve soil biology?
So, as Jamie touched on earlier, we've been using sewage sludge now for, this will be our 3rd summer. Currently on the home farm, we've been doing it, or the plan is to do it every other year for the next 4 to 5 years, and then probably go every 3 years. And then on the tenanted farm, we're going every 3 years, trying to get P&K indices up and increase nutrient availability. Also, I've got a small herd of cattle, but I've been bedding them in a corral on wood chip and straw, and then I've been composting that with waste turf and also further wood chip, and then a bit of imported horse muck. So we've probably been making about 500 to 600 tons a year, and been applying that to some of our lighter land. Just a secondary to that, we have had, which is why I asked the question regarding nematodes, we've had some issues, certainly on 20 hectares this year. I applied compost last year, and I think it's an underlying issue as well, but it's a combination of factors. I think we've got potentially some panning at depth, because it's on gravel land and it can often run into an iron pan. We've also got some BYDV in there. It's also a 3rd wheat, and we've also put compost on there, which varying degrees of turf in there, and I've got my theories that there could be fairly high nematode numbers imported from the waste turf, which is why I asked the question. So we've had some tests done by NRM, and it proved there were nematodes there at high levels, but didn't say whether they were beneficial or non-beneficial. So anyway, so to answer your question, we're using compost and sewage sludge.
Very good. Now, Jamie, let's have 3 fairly quick fire questions. One of them's a bit longer than the others, but bear with me. Well, off we go. Is a good no-till soil using chemicals better than an organic soil that's been regularly ploughed? In other words, what's most destructive to soil health, chemicals or cultivation?
Good question. So looking at research around that, again, particularly around mycorrhizal fungi, you're going to be better off with no-till and chemicals over organic. That's mycorrhizal fungi. I don't know of research looking at the entire system, but also earthworms obviously are much reduced by tillage. So if it's a difficult question to answer, but if it was my farm, I would go no-till and maybe reduce pesticides, but I would do that over the cultivations if the land allows me to do that, essentially. So I mean, always think of it as appropriate cultivations, I think, and just reduce as much as you can while still actually producing a crop and getting enough air into the soil.
Great. Okay. How would you rate chopped straw as a source of organic matter? It's likely to make a lot of money off the combine this year.
So chopped straw is a very, very it breaks down very slowly. So I've got a very high cellulose diet. So it'll sit there for quite a while and feed your microbes for a fairly long time, as it were. I think that particularly if it's mixed with sort of more accessible organic materials, it's a good part of the microbial diet, as it were. So always think of this as a mixed diet, as you would eat yourself. You know, you might have a Red Bull every once in a while, or a cup of coffee in the morning, which is, you know, something like digestate, for example, in the microbial perspective. But you'd also make sure that you have fiber and good protein, and something that takes a bit longer to break down in your body to feed you well. So that's where straw sits in that. But yeah, in general, I would recommend the more varied the diet, the better.
Okay, and I think we'll make this the last question before we do the oilseed rape poll. If crops are not hosts to arbuscular mycorrhizal fungi, what happens during the crop life? Do the amounts reduce or stay dormant?
So the amounts will reduce. It's a bit of a how long's a piece of string question, in the sense of I cannot tell you you'll start off with 100% chock full soils, and after 1 year of oilseed rape you'll have 50% left. If you think about the whole system, so if you have, for example, oilseed rape in the rotation very often, so you've got a wheat rape rotation, then you would deplete that system of mycorrhizal fungi over time, and you would probably end up with none in the soil whatsoever, particularly if you also have a lot of soil disturbance. However, if you've got very long rotations and you only have oilseed rape in there, say every 8 years, you also have other very mycorrhizal crops. Maybe you have cover crops, including legumes, things like that, and it's a lower impact system, then the impact of having 1 year of a non-mycorrhizal crop would be less severe.
Marvellous, thank you. Don't go away, we haven't finished the webinar entirely. The last part of it is this oilseed rape poll that I think I mentioned at the start. We'll close the webinar after this. So I believe some questions are about to appear, which we'd like you to answer. 4 questions, please. Well, I hope you can all see the questions. I certainly can't. Rick, Jamie, can you see them?
Yes, yeah, I can see them.
Yeah, good.
But I can't vote.
I'm supposed to be reading them out. I'm sorry, I can't see them. So I'll leave you to it for a
I'm in the background. So the first question is, what percentage of the sown winter oilseed rape area will you harvest? And we've got about 50% of you have voted. So I'm going to close that one now. So if you're interested in the results, that's the results. It's all anonymized, so it's just a percentage, but quite a mixture there. But nobody's looking, not many of you are looking to harvest 76 to 100%, which I think was kind of what we were expecting. So the next question is about spring oilseed rape, and obviously it's probably not as many of you sowing it, so it might be a bit quicker on this one. So I'll give you just a couple more moments to vote if you haven't already. And I'll close that one there. And again, as expected, 88% of you that voted haven't sown any spring oilseed rape, but again, quite an outcome for that. So the next question is about whether you plan to grow oilseed rape in 2021. And the results of that one, 60-40. 60% of you aren't planning on growing rape in 2021. And the final question is related to those 40% of you that are. It's looking at, if you are going to grow rape, what area compared to 2020? And we'll close that there, and I'll just share those results. And again, either people are growing the same or a reduced area. So thank you ever so much for voting in those polls. It just helps us get an understanding of the future of oilseed rape.
Thank you very much, Michelle. Now we'll move on to the closing bits. I mean, what I want to do first is thank our speakers. Thank you very much, Jamie. Thank you, Rick. Thank you for giving your time and all the information that you've provided us with. Thank you very much for that. Thank you, Michelle, in the background there, Michelle Nuttall, my colleague in the Knowledge Exchange team, for doing the technical wizardry and keeping things running so smoothly. Thanks, Michelle. A few bits for you. I mentioned this earlier, there's a survey just on the webinar, if you wouldn't mind completing that, please, to provide us with feedback on this one and future webinars. And a reminder that the webinar will be available, it's been recorded and it will be available on YouTube. If you would like more information, Jamie's kindly provided contact details there. There are also handouts associated with the webinar, which we've listed on your panel. There are loads of other soils resources available under the Great Soils heading, so have a look. There's loads to go at. And I just want to tell you about the webinars we have coming up. On the 24th of June, we've got the ultimate guide to the week recommended list. There are 2 chances to see that, 9 AM and 7 PM. And then on the 1st of July, we have pre-harvest preparations. Thank you very much for listening. I think that brings things to a close. Any questions we didn't manage to answer, we'll hopefully follow up afterwards. And that's it from us. So thank you for listening.
Thank you.
Thank you very much, everyone.